are the objectives in conducting this experiment. First, to be able to compare the membrane of the egg to the cell membrane. Second, to demonstrate the semi-permeability of a cell membrane using the egg membrane.
Hello friends! For today's video, I have a question. First is, are you aware of the one that protects the cell from its surroundings? Or the one that regulates water and other solute in entering the cell? If you still don't have an idea, let's learn more about cell membrane through conducting this experiment.
Here are the materials needed. Clear glass or jar, vinegar, 2 raw eggs, water, oat spoon, and food color. The next one is corn syrup, and if you don't have, you can make it on your own. And here are the procedures in conducting this experiment. Wash the raw eggs. You can put the eggs to 2 different jars, but this time, since they fit in one jar, I choose to put them together. After adding the eggs, pour a decent amount of vinegar to the jar, and wait for 48 hours to remove the shell of the eggs naturally. But take note, replace the vinegar for faster results. After 24 hours, your eggs will look like this. Take your egg out of the jar to replace the vinegar. You can notice at this point that the shell of the egg is slowly vanishing away. Pour the used vinegar to one container. Put your eggs again to the jar while repeating the procedures a while ago and wait for another 24 hours. After adding the eggs, pour a decent amount of vinegar to the jar and wait for 48 hours to remove the shell of the eggs naturally. After 48 hours, your eggs will look like this. Wash the eggs again and ready the materials needed for the next step. If corn syrup is not available, just make it on your own. Put the white sugar in a cup of water onto the pot. Mix it together till the white sugar dissolves. Add a pinch of salt and a pinch of cream of tartar to prevent it from returning to its solid form. After that, bring it to a boil and continuously stir it. After the corn syrup is made, wait for a few minutes to cool it down before using. After making the DIY syrup, pour it gently to the raw egg and submerge it. After 6 hours, take a look to the egg and you will notice that its size changes. The egg membrane shrinks. After doing the first half of the experiment on egg A, submerge it in the water mixture and wait again. After 6 hours, You will notice that the egg largens the size and it becomes more firm. On the other egg, do the opposite. First, submerge the egg on the water. Then add the food color. Wait for 6 hours, and after that, the egg will turn out to be like this. It will appear firm and full in the inside. The other half of this process is to take the egg submerged in water and submerge it in the corn syrup mixture. And after 6 hours, the egg will turn out to be smaller, or in a simple way, the egg shrinks. What happened to the eggs after submerging it into the vinegar? After submerging eggs into vinegar for 48 hours, the shell of the eggs, which are made up of calcium, becomes brittle, and in a period of time, it dissolves. Number 2. What happened to the size of egg A after submerging it into the corn syrup, or egg B into the water? After submerging the egg A into the corn syrup for 6 hours, it is appearingly seen that it becomes softer than it used to be. The membrane that covers it shrink. In biological term, what happened to it is called hypertonic. Hypertonic solution is one in which there is a greater concentration or number of solute particles outside a membrane than there are inside it. Furthermore, in egg B, which is submerged in water, shows more plump or firm structure, which is because the membrane that surrounds the egg allows water to enter. What happened to it is called hypotonic. Hypotonicity refers to a state or condition characterized by having a lower degree of tone or tension. Question number 3. As you change the submerging liquids of the eggs, did their size change? Why? As the egg B, which is submerged in water and has a bigger size first, when it is a transfer on a corn syrup, it is appearingly shows a smaller size, and the water inside the egg is released. However, after the egg submerged in corn syrup is transferred on the water with food coloring, it shows larger size than it used to be. It is because the egg membrane is semi-permeable, which allow other to pass through it and don't allow other to pass. Question number 4. What did the egg membrane portray? In the experiment conducted, the egg membrane stands as the cell membrane of the cell that acts as a guard or the protector of the cell that protects the inner region of the cell from its surrounding, which is responsible for controlling the passage of some solutes and water in and out of the cell.
괜찮아요. 괜찮아.
Ang weird naman ng hugas sa itlog. Naka-video. Visit.